Hello and welcome to the massive open online course from INFLANET. In this part of the course, I will dive into some of the cellular players in human and animal models of inflammatory crises. I will first introduce what we as scientists mean with models. Then I will give some examples of how we can model immune responses of the human body, and I will explain how certain cell culture models play a role in this. Then I will provide some examples of animal models, after which I have picked out the zebrafish model of inflammation for further explanation. I will then describe what cell types play an important role in this model, and I will highlight some of the essential cells with more information. Then I will summarize the most important information of this part of the course. So why do we as scientists love models so much? What we usually try to do when we use a model is convert complexity into simplicity, which allows for rapid hypothesis testing about the original system. What characterizes a good model is when it remains as much of the essential information about the original system as possible. Examples of models can be cell culture, animal models like mice, or more abstractly, one can think of mathematical models that are, for instance, able to simulate aerodynamics. One important thing to remember about models is that a perfect model doesn't really exist. What I mean by this is that by simplifying the model, some contextual information will always be lost, and it is up to the scientist to decide which of the information is most relevant to their hypothesis. So let's say you are interested in a specific human disease and you want to understand how it works. The first thing you can try to do is modeling this using cell culture techniques. In cell culture, specific cell types allow for specific hypothesis testing. For example, HeLa cells, which are human cervical cancer cells, can be used for studying cell division, genetics, and testing drugs. Another cell line you could use is Jurkat cells, which are human T-cell leukemia cells. These are widely used in immunological research and cancer research and are valuable for studying T-cell activation, apoptosis, and certain signaling pathways of the adaptive immune system. One of the things to remember about cell lines is that practically all of them are immortalized, which means they possess the ability to replicate indefinitely. When using cells to model and study human inflammatory responses, there are several ways of going about this. Classically, 2D cell culture models have been used. However, in these kinds of models, certain extracellular cues can be lacking, which result in 2D cell cultures being less representative of the in vivo complexity they are trying to capture. In response to this, the last 10 to 15 years has seen a big increase in the use of 3D cell culture models, where cells are grown in 3D environments, usually consisting of hydrogels or collagens with added growth factors. This increases the amounts of extracellular cues and can help cells organize into more representative structures, rather than just a flat surface. This results in models closer to the in vivo complexity. One example of 3D cell culture methods is the generation of organoids, seen here. Organoids are little balls of stem cells that with some help can differentiate and self-aggregate in 3D space to form small-scale organs. In this example, scientists have generated a retinal organoid which correctly expresses cell markers found in the human eye. Taking this one step further is with the development of microfluidic devices. Here, organoids can not only be subjected to biochemical but also biomechanical processes such as shear stress by simulated blood flow. This would capture most of the in vivo information compared to 2D and 3D cell culture, but microfluidics can be complex and expensive. This brings me to the use of animal models. The most well-known animal model is the mouse, which is used for a variety of studies ranging from genetics, disease, cancer, diabetes, and neurodegenerative disorders. Particularly helpful are genetic knockout mice, which help with identifying gene function. Fruit flies are also used as animal models for studying genetics and developmental biology. In this example, you can see an electron microscopy image of a genetically modified fly which did not develop an eye, but instead a leg. Finally, an example of an animal model is the zebrafish. Zebrafish are used for studying developmental biology, as you get eggs that can quickly develop into semi-translucent larvae. Furthermore, this semi-translucency allows for high-resolution in vivo imaging without the need for tissue clearing, which is why the zebrafish is also used for studying cell biology. In INFLANET, the zebrafish is used as a model for inflammatory responses. These can be triggered by causing an injury in the fish while they are still larvae. Most often, this injury is made by transection of the tail fin, which is able to regenerate in the course of a couple of days. However, other tissues and techniques can also be used to cause injuries, such as smaller notch cuts or, for instance, high-intensity lasers to burn the tissue. These techniques are known as sterile inflammation, which means that there are no known pathogens introduced into the response. The zebrafish model allows for the study of in vivo cell dynamics and visualization of cell-cell interactions due to its translucency, as I mentioned earlier. Using transgenic zebrafish, specific cell lines can be labeled with fluorescent proteins to visualize with fluorescence microscopy, which I will show examples of later. Zebrafish are commonly used when they are still in their larval stage, which means that at this point they do not have a developed immune system. The adaptive immunity only starts to develop around 2 weeks after fertilization of the egg. This means that in larvae the following cells are the most important ones when inflammation occurs. Neutrophils, which are an innate immune cell and the most abundant leukocyte in the body. They are the first responder in the inflammatory response. Then there's macrophages. They are the guards of the immune system and they mostly help retain homeostasis by eating dead cells or pathogens. Natural killer cells, also known as NK cells, are closely related to T cells of the adaptive immune response. However, they can act without the need of so-called self-markers. NK cells usually respond to viral infections in quick fashion. Dendritic cells close the gap between the innate and adaptive immune system as a cell that can present antigens obtained from macrophages. They present these to T cells and B cells of the adaptive immune system. Eosinophils are a cell that are employed by the immune system when phagocytosis is not an option and the problem is too big, for instance with parasites. Mast cells are well known for their roles in allergies and anaphylaxis. These cells contain high amounts of histamine, which is why if you are experiencing hay fever, you take antihistamines. I will go on to highlight the roles of neutrophils and macrophages in the inflammatory response in larval zebrafish. Neutrophils are the first responders in inflammation, as I mentioned. They can offer a variety of responses, for instance, the release of reactive oxygen species to signal for other cells, the release of neutrophil extracellular traps to capture and entangle bacteria. They can degranulate to degrade the lipid bilayer of other cells and bacteria, and they can also phagocytose to eat up pathogens or other cells. A curious ability of neutrophils is their ability to swarm to a site of inflammation. During swarming, highly coordinated movement of the neutrophils is performed, after which they can cluster together, as seen in this example. While the main objective of neutrophils after an inflammatory response is to return to homeostasis, sometimes neutrophils fail to resolve their pro-inflammatory phase. This is able to cause prolonged inflammation, and this is often associated with chronic inflammatory diseases, such as chronic obstructive pulmonary disease. The other essential immune cells in the inflammatory response is the macrophage, which are known as the cleaners of the immune system, or the guards of the immune system. Macrophages are known for their phagocytic capabilities and presentation of antigens on their cell surface. Two types of macrophages have been described. The M1 macrophages, which are pro-inflammatory and they help with cell signaling and additional recruitment of immune cells, and M2 macrophages, which are anti-inflammatory and very important in processes such as tissue remodeling and tissue regeneration. Macrophages have the same goals as neutrophils when it comes to responding to inflammatory events, which is to help the system return to homeostasis. However, sometimes macrophages can actually have adverse effects. For example, in mycobacterial infections, it has been shown that these bacteria can replicate more easily when inside of the macrophage rather than when they are outside of the macrophage. Finally, I want to show you that all of these cells that I mentioned do not work individually. Rather, they work as a team. In this video, you can see how neutrophils and macrophages move together towards the inflammatory site in the tail of a zebrafish larvae. So then, to summarize what I have talked about in this part of the course, I started off with models and I explained how they try to convert complexity into simplicity while trying to remain essential information. Then, I explained that within biology, humans can be modeled by using cell culture techniques and specific cells, or humans can be modeled by proxy using animal models. Within INFLANET, the zebrafish is widely used to model inflammatory responses. Neutrophils and macrophages are the most important cells in the larval stages of the zebrafish. And finally, neutrophils and macrophages can play either a beneficial or a harmful role in the onset of disease.